Life writes the questions. We look for the answers. Since its founding in 1896, Roche has grown from a tiny drug maker into one of the world's leading healthcare companies. From the start, founder Fritz Hoffmann-La Roche's focus was on innovation and building a company with an international presence. In the last 100-plus years, Roche has pioneered many medical breakthroughs. Our investments in research not only earned us Nobel Prizes, but also laid the foundation for some groundbreaking therapies in oncology. Today, Roche contributes on a broad range of fronts to improving people's health and quality of life. We focus on disease areas of high unmet medical need. Roche's Basel headquarters continue to be one of the group's biggest sites for research and development, manufacturing, and administration. To this day, it remains a key operational hub within the Roche Group. Every treatment starts with a diagnosis, and the more accurate the diagnosis, the better the chances of providing effective therapy. With its combined expertise in both diagnostics and pharmaceuticals, Roche is uniquely positioned to tailor treatments to specific patient populations. Drawing on advances in molecular biology and pharmacogenetics, Roche is moving closer and closer to making personalized healthcare a reality. The drug development process can be broadly divided into 6 steps. 1. Identifying a molecular drug target. 2. Identifying a first lead compound. 3. Lead optimization. 4. Preclinical safety and efficacy trials. 5. Clinical trials in man. 6. Regulatory approval and launch. 1. Identifying a molecular drug target. To fight a disease effectively, it's important to understand how people who have the disease differ from those who don't. Researchers need to understand the underlying mechanisms of the disease as fully as possible. Molecules and the pathways that influence the development or progression of a disease are potential drug targets. Step 2. Identifying a lead compound. The next step is to find a drug that will act on the target in the desired way. Basically, there are 2 options here. Researchers can look for a small chemical compound that will do the job. Most of the classic medicines of the past fall into this category. Or they can look for a large biological molecule. Therapeutic antibodies are a common type of biological molecule. Many of today's cancer medicines belong to this class. In small molecule chemistry, the challenge is to discover the right compound amid a limitless range of possibilities. The Roche Compound Library, located in Basel but accessible to Roche scientists worldwide, contains more than 1 million different compounds. And each year, medicinal chemists throughout the Roche Group synthesize thousands more. Automated high-throughput tests are often used to identify first lead compounds with the desired activity. These tests screen thousands of compounds in a very short time to identify those that interact with the target of interest. The discovery of large biological molecules follows a totally different path. An often used approach is to induce the host immune system to produce antibodies to the target. For this purpose, a living host is inoculated with the target molecule, which is recognized as foreign. The search then consists of identifying and isolating the antibody with the most favorable characteristics. It is sometimes also possible to isolate and produce a protein that occurs naturally in the body and interacts with the target. 3. Lead optimization. When setting out to find a small chemical molecule, chemists start with the first lead compound and then generate, test, and refine a succession of structurally related compounds until they come up with the optimal active structure. Wherever possible, they use computer-aided molecular modeling to better understand how active compounds and the target interact. The goal is to find a compound that has the desired effect, is well tolerated, and doesn't cause unacceptable side effects. The expression multi-dimensional optimization is commonly used because the aim is to optimize many other characteristics besides efficacy. These include the rate at which a compound is absorbed into the bloodstream and the average duration of its presence in the body after dosing. Roche researchers have access to a constantly expanding database containing detailed information on the characteristics of millions of compounds. In the case of large biological molecules, characterization, isolation, and large-scale production all are a challenge. Unlike chemical compounds, biopharmaceuticals are produced by living cells. At this point, the development paths converge. Whether small chemical or large biological, each active compound has to be tested as a drug candidate. 4. Preclinical safety and efficacy trials. Before a drug candidate is tested in man, it has to clear a number of critical preclinical hurdles. Investigations at this stage focus primarily on safety, tolerability, and efficacy. Many can be performed in cell cultures. Even in the 21st century, however, animal testing remains a necessity, for there are many questions that can still only be answered in tests on living organisms. Often, animal testing provides the first indication of potential side effects, thus helping to ensure patient safety. As far as possible, Roche uses alternative testing methods, which we are constantly refining. A new drug candidate cannot be tested in people until all the preclinical results are in and have been judged satisfactory. There are 4 phases of clinical testing in man. Phase 1. Initial clinical trials to establish safety. Phase 2. Clinical trials to establish efficacy. Phase 3. Clinical trials to establish clinical benefit. Phase 4. Post-marketing studies and surveillance. Phase 1. Initial clinical trials to establish safety. The new drug is tested under close medical surveillance in a small number of healthy volunteers to determine its safety and tolerability. These initial studies also provide information about how the new drug behaves in the human body. Phase 2. Clinical trials to establish efficacy. In this phase, the new drug is tested for the first time in patients to determine its effectiveness. Phase 2 trials are also known as proof-of-concept studies. These trials generate information on the optimal dose, dosing schedule, and route of administration. As many as 500 patients may participate in a Phase 2 trial. Phase 3. Clinical trials to establish clinical benefit. In Phase 3, the new drug is tested at a defined dose in large numbers of patients, usually at clinical trial centers around the world. It is often difficult to find patients who fill all the inclusion criteria and are willing to participate. The aim is to demonstrate that the new drug is more effective than the best currently available treatment. To ensure maximum objectivity, these trials are usually double-blinded. This means that neither the patients nor their treating physicians know which patients are receiving the new drug and which patients are receiving standard care or placebo. Up to 5,000 patients may need to be enrolled in a Phase 3 trial to obtain statistically relevant results. These trials take several years to complete and are extremely costly. The results of our Phase 2 and Phase 3 clinical trials are published on the Internet and reported in medical journals, at medical congresses, and internally to Roche employees. While trials are in progress, preparations are made to launch the product and manufacture it on a commercial scale. Regulatory approval and launch. Medicines cannot be sold in a country without approval. The authorities will demand to see all the relevant documentation, including data from all the trials and experiments conducted with the drug. In most cases, the documents supporting a new drug application would fill hundreds of binders if they were all printed out. The regulatory review process for a particular country may take months or years. If the outcome is positive, the drug can be sold in the country concerned. The leaflet supplied with each package of medicine summarizes the important knowledge gained about the medicine during the various stages of its development. In a sense, the package leaflet is a distillation of the entire research and development process. The information it contains is important both for health professionals and for their patients. Phase 4. Post-marketing studies and surveillance. Even after a medicine has been released onto the market, its use will be closely monitored. Reports of adverse events are recorded in central registries. During this phase, the drug makers often conduct additional smaller-scale clinical trials to obtain further information. Drug development is a complex process requiring tremendous know-how and a huge investment of time and money. Developing a suitable dosage form to deliver the drug to its target is part of this process. Nowadays, drug delivery systems have to be as innovative as the drugs themselves. Drug manufacturing requirements are extremely stringent. Manufacturing facilities and processes have to comply with the highest standards of quality, hygiene, and precision. Starting with the identification of a target, translating an idea into a new medicine takes an average of about 12 years, with total research and development costs of around 1 billion Swiss francs. This total includes the costs of the many projects that have to be aborted, usually because a compound proves less effective than was hoped or has unacceptable side effects. Drugs are normally patent protected for 20 years, during which time they can't be copied. But because a patent is taken out as soon as a drug clears the first laboratory hurdles, the period of market exclusivity is relatively short. Once the patent expires, other companies are free to commercialize their own versions of the drug under a different name, without having to invest any money in research. These so-called generics sometimes appear on the market within days of the expiry of the original patent. Patents are intended above all to protect inventors. A company that goes to the huge effort and expense of developing a medicine has to have a chance of earning a reasonable return on its investment once the medicine is on the market. Take that chance away, and the resources and willingness to fund research and development would soon disappear. Throughout its history, Roche has striven to be at the forefront of medical progress. To remain a leader, we are investing in some of today's most advanced technologies and steadily expanding our global research and development network. Innovation is our solution to addressing unmet medical needs. Our products are helping to relieve suffering and improve quality of life for millions of people. It takes 12 years to translate an inspiration into a new medicine. It's good that we don't give up.